Good morning, good afternoon, good evening. I'm Ara Khachaturian. Welcome to you from wherever you're joining us for episode 8 of our podcast series, Empowering Proteomics and Transcriptomics in Alzheimer's Drug Discovery. Before we start, we'd like to thank Frontiers in Molecular Mechanisms in Neurodegeneration for their continued support of these conversations, along with the International Neurodegenerative Disorders Research Center, the Campaign to Prevent Alzheimer's Disease, and the Brainwatch Coalition.
And I'm Ornit Chiba-Falek from the Division of Translational Brain Sciences in the Department of Neurology at Duke University. Today we are thrilled to host Matt Huentelmen for a conversation featuring the topic Wandering the Roads to Better Understand Cognitive Aging Across Populations, Integrating Genomics and Demographics Diversity through Lifespan into Brain Research. Matt is a renowned neurogeneticist. He joined the Translational Genomic Research Institute, TGen, in 2004, where he is a professor and director of the Early Detection and Prevention Division, and also the scientific director of the Center for Rare Childhood Disorder. His research interests center around the investigation of the omics, including genomic, transcriptomic, and proteomics of neurological trait and disease. His laboratory's overreaching goal is to leverage findings in these disciplines to better understand diagnosis and treat human diseases of the nerve system. Matt is a longtime colleague. Our paths crossed several times along our careers. Matt, welcome.
Thank you. Great to see you guys. Thanks for having me.
Matt, we're excited to have you here and to hear about your game-changing perspective with regard to transforming omics into innovative diagnostic and therapeutic solutions as we strive to prevent and stop diseases before they start and enhance, of course, obviously patient outcomes and save lives.
So this is going to take us to the first corner when we will discuss lessons from cancer genomic research into neurodegeneration. We see a lot of parallelism in multiple levels between cancer and neurodegeneration. Neurodegeneration research follows, but still tagged behind.
Yeah, I agree. I think that the way I look at cancer biology, cancer medicine, in the aspect of what I do, is through the lens of what's called precision medicine. And so about 30 years ago, when the human genome was first published, it really changed a lot of things in how we approach cancer patients and cancer treatment. And it ushered in this era of what's called precision medicine. So if we can do a little bit of history about this, I think it's really important. You know, before this era, if you had breast cancer, you were taking through a series of medicines until you responded. In some cases, you didn't respond. These medicines were prioritized based on the average response that was observed during a clinical trial. So medicine number 1 might have had a 40% chance of helping breast cancer patients, and it decreased as you progress through these. The difference with precision medicine is now to actually let the molecules within the cancer, within the tumor, tell the physician what drug the patient should receive. So instead of working through these therapies that are based on percent efficacy on average, the physician can now jump ahead, perhaps, and prescribe a medicine, use a treatment for that cancer that is more suited for that patient based on what their tumor is telling us. And so this is the concept of precision medicine. I've been lucky enough at TGen, which is really a leader in this, to see my colleagues do this in front of my eyes, to see this field mature. And I've been jealous. I want to apply it to brain disease. I want to apply it to brain aging. I want to apply it to Alzheimer's disease. And so that really has driven a lot of our areas of how we've devised our research approaches, our research projects, is with an eye towards this future, which would result in precision medicine approaches for the brain. In my case, I call this precision aging. And just real briefly, we all know there's a long list of things we should be doing to help our brain age more gracefully, right? Everything from sleep and socializing, nutrition, exercise. It's a long list. What I want to do is help us prioritize that list, help us make it personal. What's at the top of my list? What's the number one thing I can do today to help my brain? It's probably going to be different than what you guys should be doing. And I think we get there by using data, personal data about ourselves to tell us what might be at the top of our list.
Matt, that's great. It sort of actually touches on a key theme of this podcast series where we sort of entered it from this perspective of Alzheimer's and the problem of heterogeneity. And certainly this is something that the ATN criteria has come up with, trying to sort of better sort of stratify out aspects around Alzheimer's disease. Certainly, the development of genetic risk scores. A lot of this work has been, is continuing on, and it's been very important. But our interest has really been sort of thinking about how proteomics and transcriptomics can really sort of help, provide even that finer level of stratification to sort of enable, whether it's precision medicine or personalized medicine, sort of opportunities to advance new diagnostics and therapies. And so the question is, given your background at TGen and sort of from cancer research, what has cancer gotten right that Alzheimer's research operationally needs to sort of still struggling with? And is it around access to tissue or to data? Is it trial design? Where do you think that lands?
Yeah, so I think this is the really important question for us, in the field of neurology and in the field that's focused on Alzheimer's disease. So what did cancer do right? A big thing that is challenging for us right now, one of the things that cancer did right is they had a collection of medicines that were useful, that were helpful, a collection of drugs that may have been designed for different cancers, but those drugs existed and they could be used as tools. They could be understood from the molecular perspective so that in the future, when a cancer patient has a certain molecular change in their tumor, the oncologist would know this is the best drug. And equally as important, this is the drug that will not work. Let's avoid this medicine. So that's one thing that, this is an easy shot for us. We know that we struggle with medicines that are helpful against Alzheimer's disease. So that stable of medicines needs to increase. But what are things we can do today? I think we need to remember what cancer did really well in the early stages. Number one, creating big collaborative projects that just did deep molecular characterization on as many tumor types from as many patients as possible. So we definitely need data sharing. We need large studies that perform the same molecular assays and create a resource. That was really important. There's a few of those that are really critical for the cancer field. So that shared, but also very embracing the heterogeneity of cancer, which of course is a large umbrella term. We need to do the same. That's really important. I think the other important thing is to really start to understand that there's also diversity that matters at the patient level, and starting to understand how this disease behaves a little bit differently. Alzheimer's disease behaves a little bit differently, perhaps in different race ethnic groups, different groups that have different risk factor exposures. And so the take-home message is, I think we need large data sets that include standardized molecular measurements, that include very detailed clinical annotation. That was part of the big key for ushering in this era of precision medicine and cancer. We need to do the same for Alzheimer's disease.
I agree. Heterogeneity is a key word here, and we need to recognize that each of these diseases are multiple diseases, even if we're talking about specific cell region or specific organ. So let me just take a moment and talk about the relationships between cancer as a bee, as an umbrella for multiple diseases, and Alzheimer as an umbrella for multiple dementias. There are complex, often inverse risk relationship where one may influence the risk for the other. And they are coming from population-based study and also the inverse effect on dysregulation of cell cycle between cancer and neurodegeneration. Our cancer and dementia are two sides of the same coins. But on the other end, there are emerging evidence now that suggests that cancer and dementia might share a positive association. And with that, can we integrate the epidemiological link and the shared molecular risk also into the development of risk biomarker and to imply or infer from cancer to Alzheimer and vice versa?
Yeah, I think it's fascinating as well. And we've seen some recent research actually come out about this inverse relationship, uh, which is admittedly is not my area of expertise, but I think that what we need to do is start to work much more closely together across these two fields because there very clearly seems to be some relationship between cancer and Alzheimer's disease. And one of the really exciting parts is people who participated in these cancer research studies have a lot of existing data. And, maybe we can learn something about that. The other interesting part of this, though, is the known fact that some of these chemotherapies are known to induce cognitive problems that last long periods of time. So these are not just acute cognitive problems that are caused by these chemotherapies. They last longer periods of time and really can affect quality of life. That's also really interesting. The fact that we have really, and I understand it, don't get me wrong. Goal number one is to beat that cancer, right? But as we have more and more cancer survivors because of the treatments that are getting better and better, the oncologist has a better collection of medicines that he or she can choose that might protect brain function as well as helping beat the cancer. So I think that part of the tie between cancer and now generally cognition is also a really important one for us to investigate a little more deeply.
Thank you.
Yeah, so this is this is we really are sort of on the edge of a super big can of worms that we can open up, or maybe we can just kind of crack it a little bit. But certainly the topic of neurodegeneration, Alzheimer's, Parkinson's come in different forms, and certainly the rare familial and the common do not show the sort of the typical Mendelian inheritance. But then there are sort of these interesting signaling pathways, and then when you think about in neurodegeneration why cells turn off, and in cancer why cells stay on, and certainly by extension we could even be talking about other conditions like irritable bowel disorder and other viruses where cells sort of behave erratically. There does definitely seem to be a lot of commonality in terms of the biology of how these various different disorders work. And so I guess sort of a next question for sort of the second corner is like, what's your perspective? Are we looking at the same diseases? Are we looking at different diseases? Particularly within the case of neurodegeneration, is it a continuum? And what really sort of maps out sort of the common elements around cognition, behavior, and function?
Yeah, I think that's a great question. I I feel like the more we look at these different neurological diseases, the more I'm starting to be convinced that they can have a common beginning path, and then they branch out to, for example, what we see on the autopsy table, what we see, in the brain, these neuropathologies, which, I don't know if I love to believe that or not, but I think maybe it provides an opportunity to try to go backwards and even further backwards in that disease process, because maybe there is a common, or maybe just a few common ways that we end up with the neuropathology and the associated diseases. To me, that's really an exciting thing that I think we should be exploring.
So let me open here another can of worms, the animal models. So animal models today for Alzheimer's or for Parkinson and other diseases are based on familial mutation. These models are used not only for basic mechanistic understanding that we need to question, but also in translational and preclinical study. We we may cure the mouse, we may find the familiar cases using this model, but what are we making progress for the majority, but how for the majority of the patient, for the common non-Mendelian cases? Should we shift the paradigm towards other, more relevant models? And if yes, what you suggest?
Yeah, well, coming at it from a geneticist perspective, I love to simplify the genetic basis, but I think it's also really important for us to recognize the genetic complexity. So I'm going to come somewhere in the middle here. I think that definitely the monogenic models of the disease have a place, but certainly we all recognize they can lead us wrong as well. And so I definitely think that we need to spend a lot more effort on animal models that have high genetic diversity. And so I think the real power here, though, is to not ignore the monogenic models, which I know that's not what you were saying, but they need to be looked at together in parallel. They at least need to be considered side by side when we're doing some of these animal-based work.
That's great. Actually, as I was sort of thinking about your answer, my mind was drifting towards the issues of, how do we actually have sort of the means of analysis to really sort of tear into these questions and look at it. Um, just again, sort of contrasting with, say, cancer research, are there some really interesting advanced analytics that are sort of helping elucidate some of these opportunities, particularly between, say, animal models, what they sort of represent generically that could sort of map back into sort of the human species as opportunities to sort of understand mechanistically what's going on.
I don't think that there's anything, specific that's happening in cancer that, for example, we're missing on the Alzheimer's side of the puzzle. But I personally don't think that. I think the technologies that are maturing, they mature in a sort of disease agnostic space, and I think both groups latch onto them. Where we probably will struggle is, again, just due to this sort of head start, if you will, on the cancer side with deep molecular data generation. And obviously, in the age of machine learning and AI, that's important to feed those algorithms. And so, that's an important place for us to stay focused on, which is generating that multimodal, deep molecular data that we talked about at the very beginning.
Yeah, you kind of predicted where my question was going in terms of really trying to get to not only sort of the validation of, sort of more advanced modeling techniques, but also, as you said, sort of having that deep characterization. The issues of traceability and verification, we won't even get into that, but in terms of then, I guess we'll talk a little bit about this later, how to actually sort of build out those samples. So, Ornit, do you want to take us into the next corner of questions?
Before with this, within this corner, talking about genetics and the monogenic diseases. So there are also cases where single gene or variant mutation cause a severe diseases early in life and later in life are known, same genes, to affect risk for Alzheimer and Alzheimer-related dementia. Prominent examples that come in mind are ApoE and TREM2. For example, homozygosity to ApoE2 was linked to severe hyperlipidemia. TREM2 mutation, when TREM2 was identified as associated with risk for Alzheimer's disease, mutation were first identified in Nasu-Hakola disease, autosomal recessive disease that it's characterized by recurrent fracture because of bone, cyst, and also presently in dementia. Genetically, what we call like definitions, these are examples for pleiotropic. But what can we learn from that with respect to human evolution, from early age to late age? And also in respect, and this is a different aspect, to systemic nature or origin of brain diseases, and any other biological lesson that you want to give us your insights.
Yeah. So I think about this a lot because I find it to be fascinating genetically, but I also think that a lot of times people who are aware of genetics and want to use genetics, but when they approach it, they forget to remember that for something to really be showing up today in our studies, right? The genetic change has to be able to be passed on, through our many generations of human beings. So anything that has a very strong impact on that reproductive fitness, any genetic change that would be hard to be passed on, during creation of the next generation of humans, it's just not going to show up in our current studies. And so that is one of the reasons the ApoE story is a great one, right? The fact that, why would this risk factor for Alzheimer's disease, which causes these memory problems, why would it persist throughout our human story? And there's a lot of good the reason you described is a very interesting one. Its association with energy balance and things like that is also very interesting. But we also need to remember there's no negative selection for Alzheimer's disease. It happens after the age of reproduction, right? And so that's just a really important thing for us to remember as we look at these genetic findings and start to try to think about them conceptually as scientists. What do these genetics mean? I think that really matters, but I also think that the interplay or the comparison between these strong genetic changes, these things that they stick out like a sore thumb, they make an obvious protein change. And the fact that we can, we find those and they tend to cause rare diseases, right? But then we can learn about risk factors for an age-related disease like Alzheimer's is really interesting. The TREM2 story is very, very interesting. When you see it causing that disease, the disease that you mentioned, it's very clearly a mutation that results in a problematic protein. And then the change that we see in Alzheimer's disease is not that, right? But it is in the same gene, and it was informed by what we learned about these other sort of more severe, very severe phenotypic diseases that happen due to those TREM2 mutations.
Thank you. And this is going to take us to the next corner when we will discuss the early intervention for prevention. And you're heading a center that focus on that. So age-related neurodegenerative diseases, including Alzheimer and Parkinson and related disorders, start years and even decades before the appearance of clinical symptom, and even before neuropathological changes occur. So we need to start thinking about healthy brain earlier in early age groups, and treating our brain with care in midlife. We know that it will pay off during aging. So in this podcast series, we emphasize early detection, potentially by leveraging multi-omics approaches with the vision to prevent Alzheimer and Alzheimer-related dementias.
Yes. And so this is sort of an interesting topic and a little bit of a transition um, for in terms of, the work that you're doing, really trying to develop more more data. One of the issues that we sort of see happening in terms of really trying to increase um, the diversity and the breadth of different types of populations and the data is trying to marry that up with real world evidence development. And certainly there's some challenges in terms of linking up what is being collected, say, in a real world evidence environment that would be from a healthcare system level versus the needs for academic research environment. And so this is sort of one of the things that kind of really drew my attention to your work, as really trying to sort of fuse that in a way and actually come up with a way to really bring together these different types of patient populations, because I think in looking at that, that's going to be sort of the opportunity to really sort of identify more context specific signals. So as the risk benefit comes from looking at early treatment specifically for some of these invasive therapies at the pre-screening, pre-symptomatic stages, um, through the lens of a patient and provider, what might be sort of most appealing in terms of trying to get involved in this type of research? What have you seen, and what are your thoughts on this general area?
Yeah, I mean, as you can imagine, I am super passionate about this. I think that we need to do a better job of studying a larger age range of people in this world, a larger mixture of demographics. And this is a place where, in the world of neuroscience, this is a place where we have a strength. And what I mean by that is we can actually do brain testing electronically. We can test your cognition, all different types of cognition, remotely. We don't need to see you in clinic. And so we leaned very heavily into this starting in 2013 with the launch of our MindCrowd project. And I think for many years, we were looked at with skepticism, along with others who were doing internet-based cognitive testing. And then, of course, the pandemic changed everyone's mind. Everyone dipped their toes in a little bit during those years. They got familiar with understanding the pros and cons of electronic cognitive testing. And I hope that it's motivated people to start to incorporate this in a large way, as we have. And what we've seen is exactly what you mentioned. We can recruit anyone who has the ability to connect to the internet, which the studies have shown is becoming more and more democratized and more flat. In other words, so many people in this world are connected to the internet, so they can join our study from anywhere. That's really powerful. Being able to have your study open 24/7, 7 days a week, that's also really powerful. In our MindCrowd study for the past year, we have averaged over 600 new people joining our study every day, 600, which is really, really amazing, right? You could not we probably couldn't pay enough people to do that if we were requiring face-to-face visits of 600 people every day.
This is
So
impressive.
yeah, I mean, it even seems it doesn't seem real to me to even say that. When we're closing in on 1 million participants in our study, which is really, really amazing. But
I'm sorry, and the footprint here is just for Arizona alone, correct? Or is it more?
no. For Mindcrowd, we are worldwide. We're also available in Spanish, so English and Spanish. 150 different countries for the internet-based study.
That's great.
I think the other thing that's really important for us to remember is if we want to be in the early detection and prevention game, we have to be willing to follow people and study people who are currently not experiencing dementia.
Right.
And that is a really tall ask. It's really hard let me tell you, it's really hard for reviewers to put their mind around that, right? So, because those studies tend to be long, and most reviewers say, well, during this 5-year grant, you won't see a difference in these people. They're currently healthy. They're going to stay healthy. We really have to fight that. I think we need to make sure that we're focused on people who currently don't have disease and start to study them a little more deeply. For example, why aren't we studying 40-year-olds? 40-year-olds are probably the least studied group of people in this country, right? Every time I talk about studying 40-year-olds, the reviewers don't like it. They try and talk us out of it, right? But that's an important decade. 40s leading into your 50s, there's a lot of change going on there. So this is when we could have the earliest changes associated with dementia. And yet it's probably the least studied age group for when it comes to neuroscience. Really interesting.
I totally agree. Yeah.
Just
Midlife is where we're going to see the pre-symptomatic that are molecular or more omics. If we're talking about proteomics, transcriptomic changes that basically start rolling the ball and they decline.
Yeah,
what, what would
I
you
agree.
what would you tell these reviewers, or more importantly, these funders? I mean, how do we get them sort of motivated to care about the 40-year-olds midlife?
Yeah, well, yeah, no, I, I think we need to remind everybody that um, when we start to study a patient who has a new diagnosis of Alzheimer's disease, for example, to our best guess, and based on our best evidence, that's been happening in that person's brain for 20 to 15, 15 to 20 years, right? Yeah. And so why would we think that the best biomarkers of early detection would be still happening in that brain at that diagnostic state? There's still interesting things, don't get me wrong. There's still a lot we got to do there. But if we want to be in this early detection and prevention game, which we all agree we need to be, right? Because this is how you have the best chance.
For prevention.
We have to we have to acknowledge that fact, and then it's simple math to say we should be studying 40-year-olds.
I'm with you. And this is part of what we are doing here. We're starting like thinking about developing a molecular biomarker. I'm talking about omics that happened decades before, and they might be the first sign. We just need to define them and to implement it early in
Yeah,
healthcare.
I mean, this is one of the concepts that I think is it's a scary gotcha moment for neuroscience, and that is 10 years from now, will we wish we had data from 40-year-olds, including molecular blood tests, 40 and 50-year-olds? The answer is yes, So if the answer is yes, then we don't want to be those people 10 years from now wishing we could have started 10 years ago. So we got to start it, even though we may not be seeing these massive changes. We have to invest in the future. That's my 2 cents. And don't get me wrong, it's a really hard place. It's hard to sell that to a funding agency. It's hard to sell that to a reviewer that's looking at other competitive grants. But we know we should be doing that, right?
There will be a shift in paradigm.
Yeah.
I believe in us. So let me circle back to our first corner and bring again cancer as a comparison. By looking for specific DNA changes in a routine blood draw, physician could potentially catch cancer in its very earliest stage. And this is exactly, and it's also linked to the previous question, the most treatable phases. So improving the chances of a cure for more people by that. And again, we are talking exactly the parallel with Alzheimer and looking at midlife. But for Alzheimer, we can also type ApoE and maybe other rare mutation, TREM2, for example, to estimate the risk. But some would choose not to know. And what is, in your perspective, what is the readiness for implementation of early diagnosis tests in patient care, considering availability and then also accessibility to treatment currently for dementias and Alzheimer and other dementias, and the impact of quality of life by years of waiting for the worst to happen? This is also psychological concern. And what do we need to accomplish in order to get there? If you can give us your perspective from lesson from cancer, and also from learning about your CV and your career track, I saw that you also did some gene therapy development, which basically we can consider the risk between risk and benefit. So give us your perspective.
Yeah, I love this question because I think it also brings us back to kind of what's missing in neurology, right?
Mhm.
First, I think we need to acknowledge that this is a really personalized question, right? Would
Mhm.
if I could do a genetic test today that would predict your future for Alzheimer's disease, would you want it done? It is essentially, what we're asking about here. And that is really, really personal. Some people will say, absolutely, knowledge is power. I want to know my risk, and I'm going to change my life in various ways that might lower my risk or help my family prepare for that future if it looks like it has a higher chance. You could redo your finances in certain ways. You could change your lifestyle. There's a group that will say that. There's another group that's probably equally as large that will say, don't tell me about something I don't have a very clear way to completely address it and avoid it. There's not a medicine, there's not a gangbuster lifestyle change that I can do. I think those groups are probably equally sized, which is okay. This is the true human experience, but it should remind us that one of the real problems, the reason there's not one group, the reason there's two groups, is because we don't have a therapeutic solution. We don't have a drug or therapeutic approach or lifestyle change that is really convincing to people that we can address this Alzheimer's problem if it looks like their risk is higher for the future. So as we get better at that, as we have that better toolbox of therapies, these groups are going to start to merge into one, and people are going to want to know their risk. That's my opinion. I do think we see it happening in the cancer world where, yes, people still avoid some of their cancer screens like they should not do, but there's been very significant uptake in routine cancer screens. And I think that's because people know there's things that can be done about it pretty quickly.
Thank you for your insight. Ara, you will take us to the
Yeah.
last
Yeah.
corner.
The last corner, and I see we're slowly running out of time here today. But Matt, I mean, this has just been really sort of a very phenomenal discussion, learning about MindCrowd, and I'm going to direct some comments to the funders and to the reviewers out there a little bit that the work that's really being done by you, Matt, and your team is really sort of transformative, and I think it's going to definitely be paying dividends in such a way that thinking very much more holistically, perhaps maybe even really thinking across different disease orders might be the way to sort of jump that gap between people in the very pre-symptomatic or asymptomatic stages of neurodegenerative disorders, but also sort of looking at the components of other chronic disorders that could actually give rise or somehow amplify neurodegeneration. And of course, sort of this wide net that MindCrowd is really trying to throw is absolutely forward leaning, and probably will develop into sort of the 21st century version of the International Space Station in terms of generating that level of data that we need that goes across the lifespan. Perhaps you could really sort of talk a little bit about, your perspectives as thinking about not only just neurodegeneration, but a lot of these chronic disorders that span the lifespan, and perhaps just to sort of touch on how this was conceived and where you sort of see the next opportunities as technology is rapidly developing, certainly as with the development of more sources of real world evidence development, how this can sort of become more intermodally operating with MindCrowd.
Yeah, yeah, definitely. So let's talk about its conception first. I was blessed to be involved in one of the early studies in the genetics of human cognition. And during that study, this was very early in the GWAS days, and we did a study of about 1,000 people that were cognitively tested face to face. And that was amazing. That was a huge size cohort at that time. And we have a very interesting finding that's been debated, and we published and all that stuff. But the point is, as soon as we published it, we kind of knew that that was too small. We saw so much variation genetically that we didn't have a smoking gun that could predict cognitive performance on this particular memory test. So we knew right away, and we got straight to work at how do we increase this size tenfold. But cognitively testing 1,000 people face to face was already extremely expensive and exhausting, and just it took collaboration across multiple clinics to get that done. This is iPhone 2 times or iPhone 3 times, right? So you have to put your mind back in those days. But at that time, it became obvious you had to live on the internet. That was the only way we could scale what we wanted to do. I guess the other option was, to execute collaborative agreements with 100 clinical sites, which seemed like way too much, legal effort in my mind. So, living on the internet was the key to scaling. And thankfully, I think the internet's become popular, right? It has become a place where it's almost like a resource that everyone expects. We expect to be able to connect to the internet. And so that's actually really powerful. So that was kind of the origin story for MindCrowd, why we eventually made our way to the internet. I think it is really important for us to remember that we need to, just sort of back to our early discussion, we need to embrace this heterogeneity. And, coming from a geneticist, I like to try and simplify things because it makes my job easier. But we all know that's not real life. Real life is way far from being simplified. It's this complexity which we need to embrace. And it's important to do studies that result in findings. And so a lot of times we do start with the simple version, kind of to Ornit's point about, do you start with the monogenic animals, or how do you think about those monogenic animals? But the more we embrace this heterogeneity, I think the more we're going to also leapfrog and jump ahead in our understanding. I think that's another important part of this.
And stratify. So I'm sure that some of our listeners that will be excited and would envision implementing program like that, like Minecraft in other region in the US and also globally. And you already mentioned that you expand to several countries outside. But for other, do you have any tips from your experience? What to be mindful about when you start a program like this. And as we know from different initiatives, sometimes the key is beyond only launching, is sustaining and continue partnership and even to facilitate growth. So any tips
I,
for us?
I let me combine both of those and say, I think your mention of launching, this applies to all science, right? That's what we're trained to do, have great ideas. And to launch those great ideas is one part of the puzzle, but maintaining them, uh,
Mm-hmm.
realizing, what are you getting into? That's also really important to
And
consider.
keep
And
going.
yeah,
Mm-hmm.
and I think that's a really important part of this because we can all get excited about a new idea and launching a new idea. And I do this all the time too. I forget how much attention it's still going to need to
Mm-hmm.
maintain a high quality project and program. And I of course did the same thing with Minecraft, but now I'm realizing what the attention that needs, what we need to do to remain high quality. I answer emails from participants, 6 or 10 emails every day, and they're all really important questions. I didn't expect that when I launched this. I didn't think, every year I'd be answering 1,500 to 2,000 emails, about a research study. Um, so I think that's a great point. Is that and that's the advice I'd give. Have those good ideas. Make sure you follow through and launch them. But at the same time, think about this program. Is it short term? Is it a longer term program? And don't forget what it's going to take to maintain it, both effort, talent, and money. Yeah, you have to pay the bills somehow too.
Right.
So it's an oftentimes forgotten part of the business of science, I think, is that maintenance, right? That longevity of a program.
Mm. I think we're getting to our hour. Um, Ara, do you have any other question, or we can?
I do not. This was great.
All right. So, Matt, this has been a very enriching discussion. Thank you. We have talked about lessons from cancer and rare diseases research in guiding the research of the aging brain in health and disease. We discussed what it takes, where we need to be, and the challenges to enable early diagnosis as we strive for prevention and delay onset of dementias.
Yeah, this has been a fascinating conversation, really extending into the topics of population diversity on multidisciplinary aspects spanning genomics, demographics, lifestyle, and really sort of, I think, the take-home message that it's never too late to start caring for aging conditions. So, to our listeners, stay tuned for our next episode. Until then, I'm Ara Khachaturian.
And I'm Ornit Chiba-Falek. Thanks for listening.